Dear friends, good morning to all of you. I'm very happy to be with you in this lecture. And as you know, in the last few lectures, we covered the topic growth and factors affecting it. So this was the major topic of my earlier lectures. And you have realized that we need to improve the growth and yield of the growth. Simultaneously, you have seen the role of different factors, how they can influence the growth and yield of the plant. Now time has come, what should be the level of each input? How much quantity of these inputs should be applied, particularly man-made inputs, artificial inputs to realize better growth, high yield. You already know that we have control on certain abiotic factors, certain biotic factors we can control. At the same time, to realize good yield, we need to add nutrients, we need to control insect pests and diseases. So number of inputs are to be managed by farmers. So what should be the quantity of inputs so that we can get the optimum growth? So before I go into the details, these are the references for this subject. This subject I'm starting, which is basically to decide the optimum doses of fertilizer or nutrients. So you can see these are the references, Gomez and Gomez book, Statistical Procedure for Agricultural Research. Number 2, estimating fertilizer requirement. How much fertilizer should be applied to get the optimum growth and optimum yield. So this is a quantitative approach by R.D. Colwell, CSIRO Division of Soils, Canberra. Black CA or CA Black has written very good book on soil fertility evaluation and control. So all these books I will be sharing with you. Do not worry. And I'll give you some more books that are relevant to this topic as well as to this course. Now today and in subsequent lecture, what we are going to discuss or what we are going to learn. Overall, we will see fitting of response curve, how to fit it, how to find out a response curve between input and output. So you will learn in detail the difference in correlation and regression analysis. You will see how correlation, regression are different from each other. Then you will see fitting the linear and quadratic equations, models or curve. How best we can find out the line. What is the best line of fit between input and output? Then determination of optimum dose of fertilizer or optimum economic dose of fertilizer. I will discuss in detail. And what are the important uses of response curve? Because with the help of response curves, we can find out the optimum dose of fertilizer. Then the most important part will be fitting of response curves in Excel program. I will teach you online how to find out or how to fit the response curve, the relationship between input and output in Excel. Interpretation of results obtained in Excel means what is the meaning of the results that are obtained in Excel. So those will be explained, and finding the best response curve and model. So these are the topics that will be covered in coming 2-3 lectures. Now, friends, I just refresh your memories. You can see that the yield depends upon growth. I have already talked on this, and growth is affected by number of factors. Number of factors affect the growth, and these factors are genetic or internal factors. So now we are discussing with the management part. So here we should take the genotype or the varieties which are efficient, which give more yields with less doses of the input. Similarly, environmental factors like water. So we need to do proper water management, irrigation management. Atmosphere, we don't have control. N is for nutrients. Now you see, nutrients are affecting the growth. So what should be the level of nutrients that give us economic or optimum economic returns, we need to find out. And then temperature, of course, up to some extent, we can control it by mulching or by some intercropping systems or by shade net. And S is your soil. So soil management is also there, then light. So you can see that we need to find out the optimum combination of growth factors. So now, among all the growth factors, you have seen nutrients are important growth factors. And nutrients can be supplied from organic sources, they can be supplied from fertilizers, or they can be supplied from combination of both. Some nutrients can also be supplied by biofertilizers. So you can see there are different sources to supply nutrients to the crops. But the main source of supplying nutrients to the crops across the world is fertilizer. Everybody knows the value of fertilizer, that they are very, very important to increase the growth and yield of crop. But at the same time, they are expensive also. Fertilizers are expensive also. So we want to minimize the use of fertilizer. We want to optimize the use of fertilizer. Because if we use excess dose of fertilizer or more than the required, then it will create pollution in the environment, number one. Number two, it will increase the cost of cultivation. By using extra fertilizer, we are increasing cost of cultivation because it is of no use and we are unnecessarily using higher doses of fertilizer. So therefore, from 2 points of view, the use of excess fertilizer should be avoided. Number one, they create pollution. Number two, they incur extra cost. Extra cost is involved. But what will happen if we undersupply the fertilizer? There may be situation where we have not applied the required quantity of fertilizer to the crops or to the soil. So in that case, it will be called as under-fertilization or over-fertilization. In case of under-fertilization, farmer will not get maximum economic return. That is an issue. And also the growth of the crop may not be optimum. So therefore, what is there? We should not apply more doses of fertilizer or excess doses of fertilizer. We should not apply lower doses of fertilizer because we want to get more and more returns. We want to get the optimum growth. So therefore, we need to supply the right amount of fertilizer or right amount of nutrients to the soil. Now, question comes that how can we decide that this much quantity of fertilizer will be optimum? In other words, how to decide the optimum quantity of fertilizer? How can I say that this much of fertilizer should be applied to this crop under this condition? So for this, there are number of tools and techniques are available to decide the optimum quantity of fertilizer or nutrients. Friends, whenever I say fertilizer, that means I am talking about the nutrients because we are adding fertilizer to supply the nutrients. So many times I will be using these two words interchangeably. Sometimes I will call fertilizer, sometimes I will call nutrients. It doesn't matter because 100 kg urea will contain 46 kg nitrogen, which is a nutrient. So I can call it fertilizer, nitrogen fertilizer, or I can call it urea, or I can simply call it nitrogen. So now how can we decide that this is the rate of application that will give us the optimum growth? Now, number one is visual plant diagnosis. Visual plant diagnosis means you can go to the field and you find out the deficiency symptoms of nutrients. So you already know what are the deficiency symptoms of nitrogen, what are the deficiency.
Excuse me, sir. Sorry, we don't have your screen. So could you please start the screen and once share again, sir, screen?
You are not seeing slide.
Yes, sir.
Now,
Now we don't have your slide, sir.
okay. So you should have told me before.
Now,
I'll
yeah, definitely now it's sir. Okay, sir. Thank you,
no, no,
sir.
it's okay.
Okay, sir. From my side also okay.
Okay, okay. So whenever such problem come, please stop me. Okay.
Yes, okay, okay,
But
sir.
I will try to make it full. Let us see if it is getting full or not. Is it seen now?
No, sir.
No, sir. No, sir.
No. Actually, I wanted to show it in full mode, but
So now it's okay, sir. Now it's okay.
no, no. Let me open it again. Don't worry. Just a minute. Maybe I have used. I'm sorry, I closed the PowerPoint.
No problem, sir.
It is taking no, just a minute.
Very good, sir. Now we can see all
Correct.
slides, sir.
Okay, thank you. But whenever problem come, you just tell me. Okay. So friends, now I was discussing that how much fertilizer or nutrient should be applied to get maximum returns or optimum growth. How can I decide the optimum quantity of fertilizer to be applied? Now, first of all, I would like to see the deficiency symptoms, which is called as visual plant diagnosis. So in this case, I will go to the field and I understand what are the deficiency symptom of nitrogen, phosphorus, potassium, zinc. Suppose I know that these are the deficiency symptoms of nutrients in a particular crop. Then suppose the older leaves are getting yellow, older leaves are getting uniformly yellow, I can assume that it is nitrogen deficiency. I can identify that this deficiency belongs to nitrogen, phosphorus, potassium. Then after diagnosing or after knowing the problem, after knowing that which nutrient is deficient, can I apply fertilizer? Can I apply fertilizer? I can apply, but I do not know how much to apply because simply by seeing the symptoms, I cannot decide the quantity of fertilizer to be applied. That will not be scientific way. Now, next, what can I do? I can go to plant tissue test. We can take leaf, we can take any plant part and analyze the concentration or relative content of nutrients. Some procedures are available. Those procedures are called as plant tissue test. They give us general idea about the deficiency or sufficiency of nutrients. So such tests are available, which are qualitative in nature. Qualitative in nature means if you do analysis of plant samples, then you cannot know what is exact concentration of nutrients in the plant tissue. But it gives us relative idea that this particular plant or plant part is deficient in this particular nutrient. Next option is plant chemical analysis. Means this is quantitative analysis. Quantitative analysis means we can take the plant samples and we can analyze the concentration of nutrients in plant samples. And then we can see, we can match this plant chemical analysis with the critical concentration of the nutrients. Based upon the plant chemical analysis, we can see whether the concentration in our plant sample is below the critical level or more than the critical level of the nutrients. So we can compare it with the critical level, and we can know the critical level in plant or soil. But problem is that based upon critical level of nutrients, we cannot decide the exact quantity of fertilizer that should be applied. Means simply by knowing the concentration of nutrients in plant or soil, we cannot recommend the doses of fertilizer. The doses of fertilizer are difficult to recommend based upon this. Then what people suggest, if critical values are not able to decide the quantity of fertilizer to be applied, then go for soil testing. Soil testing means you can test the soil for nitrogen, phosphorus, carbon, zinc, all the nutrient analysis can be done in soil. And we can know the status of nutrients. How much is the nitrogen in soil? How much is phosphorus, zinc, or any other fertilizer? So we can do chemical analysis of the soil, which is called as soil testing. And then we can compare it with the soil fertility index, or you can call it fertility status. So we can see whether our soils are deficient or sufficient in nutrient. But again, it cannot tell us, it cannot guide us to decide the quantity of fertilizer to be applied. Then what is left? Then we can do some pot experiment. Because in the last case, in the case of soil testing, we just could know the status of nutrients, but we do not know how much nutrients can be supplied from the soil. Availability of nutrient is one thing, one aspect, but supply to the plant or supply to the crop is another point. So a soil which is having good quantity of nutrients may not necessarily supply the good quantity of nutrients to the plants, to the crops. So hence, we should know the nutrient supplying capacity of the soil. Then based upon that, we can apply the nutrients. So what people suggest, let's go for pot experiments. So we can conduct pot experiments and we can judge the nutrient requirement. But these pot experiments are not very useful. They can give us idea about deficiency or sufficiency of nutrients in the plants, but they cannot guide us to decide the rate of fertilizer to be applied, because the pot experiments are not identical to the field experiments. So then what option is left? We should go for field experiments. Field experiments, simple and complex fertilizer experiment can be conducted to decide the quantity of the fertilizer. So friends, you have seen that there are number of approaches, number of things to see whether our plants are getting adequate supply of the nutrients or not. So you can see the main purpose, the main tool to decide the quantity of nutrient to be applied to the crops is by conducting the field experiment. We need to conduct the field experiment through which we can understand the nutrient supplying capacity of the soil. We can know what quantity of nutrient can be supplied from the soil. and then whatever deficit is there that need to be supplied from outside. So this is how we decide the optimum dose of the fertilizer. So finally, I can say here that we need to conduct number of trials, fertilizer trials, to decide the optimum dose or optimum quantity of fertilizer that give you maximum returns. So this is the main area that we are going to discuss. Do you see new slide which is named as the critical level? Are you able to see it, Sayyid?
Okay, sir. Okay, sir.
Okay, it is
Yes,
moving
teacher.
now. So now we need to understand, last slide I was talking about critical level. We can analyze the plant samples for nitrogen, phosphorus, potassium, or other nutrients, and we can see what is the concentration of the nutrient. So that concentration can be matched. Matching can be done with the critical value. Here I want to inform you that critical level of nutrients are already decided. Many people have worked on this and the values are available. Standard critical concentration for different nutrients in different crops are available. Data are available in every country, every place. So we can compare our result with those standard values. If our results or values are lower than that or lower than the critical values, then definitely we need to supply the nutrients. But really it cannot help us to decide the exact quantity of nutrient to be applied. That can be decided only by conducting field experiments. We can decide the optimum rate of fertilizer. So here now, I just, you can know what is a critical value. What is the critical level or critical value of the nutrients? So dear friends, this critical value can be in soil or can be with respect to plant. So it is the level of concentration of a nutrient in the plant or in soil that is likely to result in 90% of the maximum yields. Suppose there is no deficiency of any nutrients. All nutrients are available in sufficient quantity in the soil. So then you can see the plant will give you, or crop will give you maximum yields. So maximum yields are obtained when all the nutrients or all the essential nutrients are present in right and adequate amount in soil. So that is 100% yield is obtained. But when you get only 90% of the maximum yields, that at that particular point of time, the concentration of nutrient in plant or in soil is called as critical concentration. Critical concentration is the concentration of nutrient in plant or soil at which plant gives 90% growth or plant gives 90% yield. So that concentration in soil or plant is critical concentration for deficiency. Remember, this is for deficiency. Similarly, sometimes certain nutrients, particularly micronutrients, iron, manganese, zinc, copper, they can be accumulated in much higher concentration than the optimum level. So those kind of elements or nutrients can cause toxicity. So there are two extremes. One extreme is deficiency, when nutrient levels are insufficient. Another level of nutrient can be sufficient or more than sufficient, or it is in excess. Then it will cause toxicity. So micronutrient like iron, zinc, manganese, copper, they can result in toxicity to the plant. So their concentration of these toxic elements at which they start damaging the crop is called as critical concentration for toxicity. So you can see it can be in soil and it can be in plant. Is it clear, Javed? This critical level? Nabi? Okay, Bismillah, are you?
Yes, sir.
You could
Yes, sir.
understand?
Yes, sir. It is okay.
Okay, critical concentration. Okay. Now you can see more information of the critical concentration. So the range of nutrient supply from deficiency to toxicity. Deficiency means the nutrient in soil is present in very low amount, and toxicity when nutrient is present in very high amount. So you can see this picture. It shows plant growth and yield. It depends upon nutrient supply. So nutrient supply is a growth factor. So now on the x-axis, you can see this is the concentration of nutrient or nutrient content in plant or in soil. So it is increasing from left to right on x-axis. Now see on y-axis. On y-axis, it is the growth or yield which is increasing. From lower side to upper side, the yield or growth is increasing. Now see initially on x-axis, the nutrient supply in soil or nutrient content in plant is very low. So because of low level of nutrient in soil or plant, it is giving you acute deficiency. Acute means very severe deficiency. But you still increase the supply. Now you will have more nutrient if you move from left to right on x-axis. If you increase the supply, then you can see deficiency is still there. The nutrient deficiency is still there, but it is hidden. You cannot see it, but plants are suffering from shortage of nutrient supply. This is known as hidden deficiency or hidden hunger. So in this zone lies the critical value. Critical value, if you move towards left side, so here you will get 90% growth or 90% yield, or in other words, the reduction in growth or yield is 10%, 10% or more. Then this is your critical value. And then after critical value, you continue to increase the nutrient supply. And then one point will come, which is optimal supply. So we continue with the same picture. And you can see after optimal supply, certain nutrients are accumulated in excess, which is called as luxury consumption. Luxury consumption here means it happens for potassium, which is accumulated in high amounts, but it is not toxic. This potassium is never toxic, even if it is accumulated in high amounts. But it will not increase the yield. The yield will remain same, you see, under optimal supply and luxury consumption. You can get 100% yield, 100% yield, but it is wastage of nutrients. And then if you still continue to increase the supply of the nutrients in the soil or in plant, then the toxicity of the nutrients will happen and it will reduce the growth. So you have seen different supply level and their effect on the growth. Whether a particular nutrient is deficient or it is in excess, it can lead to reduction in growth and yield of the crops. Now you can see the critical levels or critical limits in soil and plant tissue. Fully developed youngest leaf for micronutrient deficiency in field crops. So you can see for zinc in soil, the extractant is DTPA. DTPA is an extractant with the help of which we can extract zinc. We can extract the zinc from soil or plant. Then the concentration in plant is 0.6 microgram per gram. This is 0.6 microgram per gram, or you can say it is 0.6 milligram per gram, milligram per kg. Equivalent units are there. You can call it 0.6 ppm. You can call it 0.6 milligram per kg, or you can call it microgram, 0.6 microgram per gram. Similarly, plant tissue critical limit in microgram per gram or ppm is 10 to 20. So 10 to 20 is in plant tissue. Means if your plant is having 10 to 20 ppm of zinc, then it is deficient in zinc. Similarly, for manganese, DTPA is the extractant, and 2 ppm is the concentration of critical limit in soil. Means if your manganese in soil is less than 2 ppm or less than 2 microgram per gram, then it will be considered as deficient. In plant tissue, similarly, if it is 15 to 25 ppm, it will be considered as deficient in plant also. Next is iron. Iron concentration in soil is 2.5 to 4.5 microgram per gram is critical concentration, and in plant tissue it is 50 ppm. Similarly, boron can be extracted by hot water, and its critical limit in soil or critical concentration is 0.5, means it is deficient, and then 5 to 30 ppm in plant is deficient or critical concentration. Now, friends, you can see methods for evaluation of soil fertility. I was talking, or I was saying to you that we need to test the soil. We need to test the soil to know the exact status of the nutrients. So there are different methods for evaluation of soil fertility. We have already discussed. I'm just repeating them here. So first of all, I told you that soil fertility can be evaluated, or deficiency symptoms or nutrient status of plant or soil can be judged by visual plant diagnosis. Visual means just seeing the deficiency symptoms. So symptoms first appear on all leaves. We know it is in case of nitrogen, phosphorus, potassium, and in case of certain micronutrients like boron, deficiency boron, iron, deficiency symptoms come on younger leaves and not on older leaves. Then we can go for plant analysis. I told you it is also an approach to supply the nutrients. So in this, there are two approaches. One is rapid tissue test, which is a qualitative test. It gives us just idea or indication that our plant tissues are deficient, but it will not give us exact concentration of the nutrients. Then we do quantitative analysis. Quantitative analysis is total analysis of nutrients in the plant. So this is total analysis. Bismillah, do you have facility for chemical analysis of soil and plant in your lab? Anybody? Do you have facility?
Sorry, sir, we don't know about your question.
Okay. But you should try to establish a lab where you can have minimum facility for nitrogen, phosphorus, and nutrient analysis, soil analysis. This is very important. This is more important than variety development or plant breeding. So you may think of establishing a soil fertility lab where you can have some modern equipments, and you can see behind me there are some instruments kept here. If you can see it, this is the lab. I'm not able to show you complete, but this is the lab where I am associated and doing work here. So you can also think of establishing a lab. Then, so you can do total chemical analysis for nutrients, and in this case,
Yes, the slide is visible, sir.
okay, fine. Now you see we were doing discussing about the total elemental analysis of nutrients. So in this we need to sample the plant sampling is there, and after plant sampling we need to process it. You need to dry it, grinding is done, and then sample is ready. Sieving is also done, and then you can extract the nutrients from the sample by putting some acids, sulfuric acid, etc. You extract the nutrients and then analyze them. And finally, you go for interpretation. You can know the critical concentration, you know the nutrient ratios, risk system, and chlorophyll meters and LCC, etc. This part will be covered in soil fertility course, so you need not to bother about it. Now, soil testing or soil analysis. In soil analysis, what we do, we go for soil sampling, labeling, processing of soil samples, and then we extract the nutrients with the help of certain extractants, and then we do the chemical analysis of the nutrients in soil. And then we go for interpretation of nutrients, and this will be taught to you in soil fertility course. But here you can just see that these are the different methods for evaluation of soil fertility. Now then microbiological methods are also available, and then remote sensing methods are also available. And finally, you can see field experiment. Field experiments are to be conducted to decide the optimum quantity of fertilizer that should be applied to get the optimum yield. Now, field experiment like controlled test plot and complex fertilizer experiments. So in controlled test plot means it is very simple. You can get a field or two fields. You can get say 50 meters square each or 100 meters square each. In one plot, you do not apply fertilizer. In another plot, you apply fertilizer. So this is a simple fertilizer experiment where you can show response of fertilizer to the farmers, to the people. Okay, in one plot, I have not applied fertilizer. In another plot, I have applied fertilizer. So you can compare the results, but it will not tell us how much nutrient exactly to be applied. For this, we need to do further analysis. And then there are complex experiments. In complex experiment, you can take two different nutrients. Instead of nitrogen or in addition to nitrogen, I can take phosphorus also. So two sources or two nutrients can be taken. So whenever you have more than one variable or more than one nutrient or more than one nutrient source, it becomes complex experiment. So two kind of experiments, field experiments are conducted. So finally, you can see to decide the optimum rate of fertilizer, you need to conduct the fertilizer experiment. So it steps in interpretation of results obtained by conducting fertilizer experiment. So overall, whenever you want to decide the optimum dose of fertilizer, you need to conduct the fertilizer experiments under field conditions. And you will apply different doses of the fertilizer, say 0, 40, 80, 120, 160, 200. So different doses of the fertilizer will be applied by you to the field. And then you will record the yield data. The economic yield will be recorded. That what was the yield at 0 kg, what was at 40 kg, 80 kg, or 120, 160, 200 kg. At different levels of nitrogen or any nutrient, you need to record the data on the crop yields. So you can see the results are obtained. So you will get the results with respect to growth or yield. You can have several parameters. You can have growth in terms of leaf area index. You can measure plant height. You can measure number of leaves per plant. You can measure root dry weight. You can measure dry matter production. So different variables can be taken or can be recorded on the plant, which is called as dependent variable. The variables whose outcome is dependent upon other variable is dependent variable. See, here you have applied nitrogen fertilizer. So nitrogen fertilizer is an independent variable. It is affecting the outcome. It is affecting the outcome. So this is actually the cause variable. This is actually the cause, C-A-U-S-E, cause variable. And the other variable is effect variable or response variable. The Y variable or dependent variable is response variable. So that Y variable can be leaf area index, can be dry matter, can be plant height, can be grain yield, can be panicle length, any dependent variable you can take. So here, there could be one dependent variable, that is grain yield, and other one independent variable is fertilizer rate or rate of fertilizer application. So you record the data on the grain yield. Then you know whatever fertilizer doses you have applied and respective yields have been recorded. You have recorded yield for control, yield for 40 kg, 80 kg, 120 kg, and all the levels you have recorded the yield. So first data is say X0, Y0. X0 is fertilizer is 0. So whatever yield I get at X0, I can write it Y0. Similarly, X1, Y1, X2, Y2, X3, Y3, I will have the data of independent and dependent variable in pair form. The data will be in pair form, or data on independent and dependent variable, or you can say fertilizer rate and grain yield, maybe in pair. That will be pair data. Now you go for statistical analysis. I do not know whether you understand F-test or test of significance or analysis of variance. Analysis of variance, do you understand, Rahim? Analysis of variance, do you understand? Rahim?
Yes, teacher, yes. We know
This statistical
about variance analysis.
analysis of variance. So here you see, you have recorded the data, the grain yield at different level of fertilizer. So now there may be variation in yield. The yield which is obtained at control may be different from yield obtained at 40 kg, 80 kg, 120 kg, and so on. So yields are not same. There are variations in the yields. So why this variation has happened? Why this increase in yield has happened? And sometimes a decrease can also happen. So what are the reasons for it? So we need to analyze those reasons. We want to see the reasons for this variation means variance. So analysis of variance means it partition different components of variation. Which particular element has caused variation in the yield? Who is responsible for variance in the yield or in the data? Just a minute. टॉपिक क्या है? काम का ही है, वही होगी सीधी वो है किसमें? Yeah, what? ठीक है sir, ठीक. So you want to ask something? You raise hand. You are welcome to ask question.
I think, sir, he touched the icon. There is no question, I think.
Okay, no problem. So you see, now you understand that different sources of variation are partitioned into different components. For example, one source of variation could be replication. One source of variation could be replication because you record data from different replication. First replication, second replication, third replication. Each replication will have equal level of treatments. Suppose 9 treatments are there. So 9 treatments will be repeated 3 times. So that is your replication. So source of variation can be replication because fertility of blocks or different replicates may be different. So due to replication, you can get variation in data. Second source could be treatment itself. Treatment itself means you have applied different doses of fertilizer. So this may have caused variation in the data or increase in the data. So second source is this one. And third source can be error. Error, because there may be some unexplained reason for the variation in the data. That becomes part of error. So, and then you have total variation, total variance. and out of total variance, how much is going to replication, how much is going to treatment, and then remaining will be deducted. The deducted part will be error. So this is how we go for statistical analysis, which is called as ANOVA. It is done by F-test. So F-test tests the variance. And then it tells us whether the results are significant or not. So it tells us whether the results are significant or not. If our results are significant, means the calculated value of F is more than table value of F, we say or we declare that the results are significant. Table value of F at 5% can be seen. It has different degrees of freedom. In case of P, you have only 1 degree of freedom. But in case of F test, you have 2 degrees of freedom. One you need to see horizontally and one you need to see vertically at the prescribed level of significance. Then if your values are greater than the table value, then results are declared significant. That means the variation or increase in yield is actually caused by the treatment. It is because of treatment. It is because of increasing doses of fertilizer that our data are significant. So once our results are significant, we go for correlation and regression analysis of data. You have data on input, you have data on output. Then we need to go for correlation and regression analysis. If our results of F-test or ANOVA test are significant, if our results are non-significant, then we cannot do further analysis. We stop here. So once our results are significant, we go further. If these results are non-significant, means calculated value of F or F test is less than the table value, then our results are non-significant. That means we cannot proceed for correlation and regression analysis. Since suppose here results are significant, then we go for correlation and regression analysis. You will see how we can do correlation and regression analysis. So through this correlation and regression analysis, we will find a response equation. And from this equation, we can do predictions. We can estimate the optimum dose of fertilizer, and we can make a lot of predictions or forecasting from that particular equation. And that some economic principles can be applied to the response equations, and these equations are also called as regression equations. So certain economic principles can be employed to these regression equations, and some mathematical relationship can be developed to find out the optimum dose of the fertilizer. For linear, suppose relationship is linear, then we cannot find the optimum dose of fertilizer. But if relationship between input and output is fertilizer, output is grain yield. If the input and output relationship is quadratic, then we can determine the optimum dose of the fertilizer. So friends, here you can see that the knowledge of 3 subjects is combined to understand it. Agronomy, how agronomy is involved? Because we need to conduct the field experiment. We have conducted field experiment. We have used different doses of fertilizer, and we have recorded the yield of crop at different doses of fertilizer. Then statistics come into picture. We have done F-test, or we have judged the significance of the treatment, and it was found out to be significant. And then we need to find out the optimum dose of fertilizer. So some economic principles are to be employed that you will learn subsequently. So overall, you can see that knowledge of 3 subjects is necessary to understand the economic analysis or when we decide the optimum dose of the fertilizer. Any question at this stage? Any question at this stage?
Thank you very much, sir. No question from our side, sir. Please continue the lesson, sir.
So now, friends, if you understand that, now we go further deep into the subject. This is response curves. So as I told you, response curves help us to decide the optimum dose of fertilizer. So how to define it? Functional relationship between input and output is a response equation. Okay, functional means mathematical relation between input. So here input is fertilizer, and fertilizer is a kind of independent variable that is called as independent variable. And then output is our grain yield. So grain yield is our dependent variable, because grain yield depends upon the rate of fertilizer. If you increase the rate of fertilizer, grain yield will increase. So therefore, it is called as dependent variable, and fertilizer as independent variable. Relationship between yield and fertilizer rate is called as fertilizer yield function with a general mathematical form or model. Friends, these models, equations, curves, they are same thing. There is no difference. Some people call them curves, some call them response curve, production functions, or models, this and that, but they remain the same. The thing is same. It is only the way people call them. So for same equation, you can call model also. So model or equations are estimated from experimental data will give us a relationship, realistic mathematical relationship of the biological relationship indicated by the data. So we can develop model, we can develop equations to establish the relation between X and Y. X is independent variable and Y is dependent variable. Now you can see variety of response curves can be there. In the first column of the table, you can see name of the degree, name of equation. Equation can be also referred as model, and then name of the curve. You already know that if you got one equation, I know that the algebra, you know, mathematics is, Afghani people are very good at mathematics. I realize many times. So you understand this subject very well, I hope. And you see the models, different models. First degree model. First degree means power of X. This is power of independent variable. So X power 1, X power 2, X power 3. Then name of the equation. If power of X is 1, then the equation will be called as linear. And the name of the curve will be a straight line. Then second equation is quadratic equation. Here the power of X is 2. Then the name of the curve is parabola. Then there could be third degree. Power of X means independent variable is 3. Then the curve will be cubic parabola. And then fourth degree equation will be, it is quartic. It is quartic, friend. Make it quartic. It is not quadratic. Q-U-A-R-T-I-C, quartic. And the name of the curve is also quartic parabola. It is wrongly written quadratic. You make it quartic. Q-U-A-R-T-I-C, quartic parabola. Then fifth degree is quintic, X power 5, and it gives us quintic parabola. So a production function can also be called as response function, is a mathematical expression that describes the physical relationship. It shows us only physical relationship between input and output, just physical relationship. The relationship is expressed as y is equal to function of x1, x2, x3, and xn. So friend, here y is output or y is yield, and y is also dependent variable. It is also dependent variable. So it is function of several independent variable. Number of independent variable will affect the dependent variable. For example, yield can be affected by several nutrients. It can be affected by nitrogen, phosphorus, potash, and so on. Similarly, these variables can be herbicide, pesticide, or fertilizer doses, or water quantity, or so on. So you can see yield is affected by number of factors. So it is difficult to study effect of all the independent variable on the dependent variable. Very tough subject, but we can study one. If we have one independent variable and one dependent variable, then their explanation becomes much easier. As you increase the number of independent variable affecting a one dependent variable, then the interpretation becomes complex. Now you can see the type of response curve. So here linear is a straight line, you can see. So it is negative correlation. This is quadratic kind of structure, you can see. This is cubic. Line is like this, and then quartic, and then exponential regression. So these are different shapes of the curves. Now see what are the types of equations or types of curves. The nature of equation will decide the nature of curve. A quadratic equation will give us quadratic curve. So therefore, equation is just graphical representation of the data. Now you can see what is exponential equation. So there are two kinds of equations. One is exponential equation, other is polynomial equation. Exponential equation means the exponent means power of variable. The exponent or a part of it is a variable. For example, 3 power x. So here you can see x is variable actually, but it is in the power. Then suppose 3 power x is equal to 81. Similarly, 5 power x minus 3. So these are actually your exponential equations. Or you can simply say e power x, or e power minus x, or e power minus 1 by 2x. So wherever exponent variable is the exponent or power, then it is called as exponential equation. And then polynomial equation is an expression consisting of variables, maybe x, y, w, z. Coefficients, here coefficients are to be there, b and c. And then exponents of 1 and 2. Means here powers are 1, 2, 3, 4. Powers of the variable. And polynomial function is one which has a single independent variable. So here there is single independent variable. Y is equal to a plus bx power 1 plus cx power 2. So this is a quadratic equation. Here y is dependent variable and x as independent variable with degrees 1 and 2. Here you can see bx1 and cx squared. So degrees are 1 and 2. Highest degree is 2. Here the highest degree is 2. Thus it is called as binomial or quadratic equation. Similarly, you can have cubic or quartic or quintic, any other equation. So exponent in a polynomial equation, P means polynomial equation, can only be a positive integers. Natural number, it is always 1, 2, 3, 4. Negative integer exponent is never a part of polynomial equation. The independent variable x can occur multiple times. As you have seen in the last case, it was twice. Different x's are separated by addition, subtraction, or multiplication. The degree of polynomial is highest degree of the exponent that exists in the equation. Now, you see polynomial of degree 0 for a nil relationship means y is equal to 0. There is no relationship. If you have polynomial of degree 1, then this is linear relationship or a straight line, y plus bx. Now you see if polynomial of degree 2, it is a quadratic type of curved relationship, y is equal to a plus bx plus cx squared. Polynomial of degree 3, it is cubic type of curved relationship, y is equal to a plus bx plus cx squared plus dx cubed. So here you can see there are 4 terms. So dx cubed. Polynomial of nth degree, y is equal to b0, b1x, b2, x squared B3x cube plus Bn x power n. So this is polynomial of nth degree. Means it could be for 3, 4, 5, 6, 7, 8, 9, 10, whatever you want. So these are different types of equations and curves. So polynomial of degree 1 is a linear response. It is a straight line. Polynomial of degree 2, 3, 4, 5 up to n are curvilinear or non-linear. They are called as curvilinear or non-linear in nature, representing version of the law of diminishing returns. Economic interpretations are made from these functions. So once you have found out these kind of relationships which are non-linear, so from non-linear relations, we can work out the economic relations, or we can work out the economic optimum dose. Economic interpretations are made from these equations. Procedure for fitting different response functions to experimental data are available. Now, for fitting different response equation means to find out the relationship, and that relationship can be represented by equations. So procedures are available, and procedure is method of least square that we are going to discuss in the subsequent lecture. How to fit an equation and what does it mean all. We will discuss it. Procedure for applying economic principles. I think only 1 minute is left. So we can break here. So any question up to now?
No, thank you, sir. No questions.
So friends, we will meet after 15 minutes.
Okay.
I'll type a message on the WhatsApp to join. I want rest for 5
Okay,
10
sir.
Okay.
Okay, sir. We will join the third link. Yeah.
Yeah, third. And I will
Well,
type
just-
message. I will type message in WhatsApp to join.
So just text us that we will be joined. Text us.
Thank you. Thank you.
Yes, thank you, sir. Thank you.
So dear friends, welcome back in this class. And if you see in the last slide, you have seen different types of equations or curves. Now, procedure for applying economic principles to experimental data via different response functions generally worked out by the individuals. What is the message out of this sentence is that once you have generated the equation, once you have fitted the equation, or once you have found out the equation, then you can apply economic principles to generate a result on economic return. You want which is the best level of that independent variable where you will get maximum returns. So such economic interpretations can be made from these equations. The linear response implies constant return. In linear response, you know, it is a straight line. So you get constant return. That means with the addition of each unit, you get the same. You add 1 unit, you will get the same output. Second unit, same output. So output with the addition of each unit remains constant. So application of economic principles to this function is different from and simpler than the applications to the curvilinear diminishing return equations. Friends, all these equations can be divided into two groups. One is linear, straight line, and other is called as non-linear. And non-linear is having polynomials of 2 or higher degrees. And these non-linear are also called as curvilinear relationship. They are also called as curvilinear. Curvilinear means there is some part linear, but overall they are not linear. They are curvilinear. So curve is there and linearity is there. So both are there in curvilinear or non-linear relationships. Now, friends, now question comes, how to find out the equation? How to develop the equation? Development or to find out equation is known as response curve fitting. Response curve fitting means you have two kinds of data. One data is independent variable, which is in this case a fertilizer dose, or you have used fertilizer doses, suppose here 30, 60, 90, 120, 150. You can see on x-axis, different fertilizer doses are there. On y-axis, suppose these are the yields of potato in tons per hectare. On y-axis, you have those yields. So here on y-axis, you have dependent variable. On x-axis, you have independent variable. And then you can see here that the data has been plotted. This data you have got by conducting the experiment. You have conducted one experiment where you have used different doses of the fertilizer starting from 0, 30, 60, 90, 120 and up to 210. And then at each dose, you will get the respective yield. For example, at 30 kg fertilizer, you get this much of yield. At control, you get this much blue, you can see this blue solid, this. And then at 90, at 60, you got this. At 90, you got this point. At 120, this point, this point, this point. So all the data have been plotted on the curve, on the graph paper. And if you see just this data, this is called as a scatter plot. A scatter plot where data has been plotted for input and output, or for independent and dependent variable. So now you see these data point 1, 1, 2, 3, 4, 5, 6, 7, 8, 9. There may be 9 data points. So now we cannot join these points by hand. We cannot join these points by hand because we do not know what has happened from 30 to 60. We have no authority, legal authority, to join these points by line. We cannot join them. So what can we do? We can find a line. We can find a line that best fits this data. We need to find a line that represents this data the best. So we need to find the best line of fit that represent this data the best. So you can see many lines can be made. Many lines can be made. This just I made randomly without any consideration. I made this green line. Okay, this can represent this data better. Then I put purple color line. This can represent. Many lines can be drawn to represent this data. But which line is the best fit? Which will, how to decide that this particular line will be the best fit or will represent this data the best? To find out the best line fit, we need to use the method of least square. Method of least square means the line which has least sum of square of distances in observed and predicted data. This line will give us predicted data. This is this purple line, black line. These are predicted data have been put on these lines. So method of least square, you can take the distances. Suppose this is purple line. So you can take the distances. Some data may be above this line. Some data may be below this line. You can see below this line. Some data are above the purple line. So you can measure the distances. You can measure the distances, count the distances, measure the distances, and square each value. Square each value and take the total sum of square. The line which is giving us least value of the total sum of square of the differences is called as best fit line. Let us understand more. You can see no straight line passes exactly through all points, though many can be drawn. You have seen that many lines can be drawn, but no line passes through all the data. No line can pass through all the data. Find a straight line that fits all points better than all. So we want to find out of these lines, which line will give us the best fit, which line can represent this data the best way. In other words, what value of A and B in the equation y is equal to A plus bx must we take? because linear equation is y is equal to a plus bx. So y value, we are going to have relation between y and x. So here, if you know the value of a and value of b, we can fit the line. We can fit the line. So here the question comes, how to calculate the value of a and b? Can we devise a measure of all closeness of the fit of the various lines which can be drawn? It is also possible that data follow diminishing kind of curve whenever non-linear regression would be better fit. So it is always not desirable to fit the linear curve or linear line. Sometimes our data may be suitable for quadratic or some polynomial kind of relationship. So in this case, first of all, we need to plot a scatter plot. And this scatter plot will give us idea whether this relationship could be linear or curvilinear. Then response curves are fitted by using method of least square. So we need to understand what is method of least square. Based upon this method, we can find out the best fit line. Now, let us know more about the method of least square. It is a statistical method used to determine a line of best fit by minimizing the sum of squares created by a mathematical function. So sum of square of distances between observed data and predicted data will give you the best fit line. A square is determined by squaring the distance between a data point and the regression line. You will learn it. You will learn it in the next slide. I try to explain more. So here you can see 3 lines have been drawn here. Number 1, on x-axis you have independent variable fertilizer doses. On y-axis you have grain, suppose. So y-axis you have, this is your dependent variable, which is yield. On x-axis you have fertilizer, which is independent variable. So you know the data is in pair. At 0 value of x, you will get some value of y. This is suppose x1, this is y1, x2, x2, y2, x3, y3, x4, y4, likewise. So you can have data in pairs. Similarly, in the second graph and third graph. So what is required here? This is the line. Suppose I assume that this is the best fit line. I assume. And from here, we need to calculate value of A and B also. So here, what you can do, this is the line, and this is the predicted line, and this is the actual data. So actual data minus this data. Actual data minus this data. Suppose this is 3. This may be, say, or this may be 4.8. 4.8 minus 3. So you can find out the value and square this value. Square this value and keep it to make the total. Similarly, next value. Next value will be positive because observed value is more than the predicted value. Predicted value is on this line. So observed data minus predicted data. This is the distance. So you square this value and keep it for total. Then similarly, next data, this minus this. This distance, square the value, this distance, this distance, this. But the data which is below the line will give you negative values because this is observed data, actual data, minus predicted data, this one. So this minus this will give you negative value. Therefore, we need to square these distances. So here we can square it. And then again, the last, this data is your predicted data. This is your actual data. So this minus this. And then whatever is sum of squares are there. These sum of square may be totaled. You need to make total of all these sum of square. Similarly, let us take another equation. This line, I assume that this is the better fit. So same operation I will do here. This value minus this value minus this value minus this value, and square every difference. Square every difference and make the total of all. Similarly, for the third line, you do the same exercise. So here also there will be total sum of square. Total sum of square of differences of observed and predicted values. So this total of sum of square here from this equation, from this equation, from this equation, or from any other equation. Our equation will be the best fit where we get the lowest value of the total sum of square. Or you can say the value should be least or minimum. Then therefore it is called as method of least square to find out the equation between independent and dependent variable. This is for linear relationship. Any question, any doubt in this? Rahim, Nouri, Bismillah, Hikmatullah.
No, sir, thank you. It is okay.
No question, sir. No
Okay.
question. Thank you.
I'm happy. Now see, you can understand more how we fit this equation. So you see, x-axis same value, y-axis is your yield, and here you can see for every the black data, y1, y2. This is in capital letter. This is in capital letter. This is your observed data. This data you have recorded from the field. And the green line, this data is predicted data. So now what you can do, y0, capital Y0 minus small y0, capital Y1 minus small y1. So these distances, and square each difference and make the total. So that total will give us the least or minimum sum of square. So therefore, this line would be the best fit. So this can be written in the table form. So one is output. This is your dependent variable. And here on the left side, you can put independent variable. x0, x1, x2, x3, x4, and xk. Then output is y, dependent variable. Y0, it is when x is 0. Y1 when x is x1. Y2, y3. So these are your actual data from the field. And suppose on the equation, on the line, you have predicted data or expected data represented by a small y. So it is y0, y1, y2, y3, yk. Similarly, now you take sum of difference. Sum of difference means this minus this, this minus this, this minus this. So we can subtract it. And next column, it is not given here, but you can square all these values. You can square all these values and make the total. Because here differences may be, some value may be positive, some value may be negative. So therefore, you need to square individual values and then make the total of all. And this total should be the least or minimum. Therefore, it is called as method of least squares. Now, there are some residuals. Residuals means here error. Some data remains unrepresented. See, difference of these two. The difference of these two is your residual. The difference of these two is your residual. Means this data, residual data, is not represented by the equation. So this is called as residual or error. In regression analysis, error is difference in observed value, y value, and predicted y value, small y. So it is actually distance of a point from the curve. How far is the observed value from the curve? That is called as residual or error. Residual is positive when point is above the curve, and it is negative when the point is below the curve. I've already told you. Now you can see here, this is your actual data, observed data. If it is below the line, then you will get negative. The difference in observed data and predicted data will be in minus. Therefore, you need to square these values. Similarly, the data which is above this line, it will have positive residual. So square these values because this line is not exactly passing from all these data points. The best thing will be if this line is able to pass through all the data points. But that is not possible because there are some errors, there are some residual. So you know on x-axis it is x, on y-axis it is dependent variable. Hope you understood the meaning of residuals. Means the distances from the line. Now, friends, before going into the details of response curve fitting, I deem it fit to explain you what is meant by correlation and regression. So I will go into further details of this response curve fitting. But before that, I refresh your memories and what is correlation analysis and what is regression analysis. What is difference in correlation and regression and correlation and regression coefficients. So you will learn all these techniques now. So correlation analysis provides a measure of the degree of association between the two variables, whether the two variables are related or not. If two variables are related to each other in one way or the other, then it is called as correlation. Or you can say it is a mutual relationship or connection between two or more things or two or more variables. Correlation is a statistical technique that can show whether and how strongly pairs of variables are related or not. So first part of correlation analysis is just to judge whether two variables are connected, whether two variables are related to each other or not. If they are related, then how deeply they are related. Okay, me and Nouri are friends. Okay, we are friends. We just shake our hand, but we are not deep friends. It is never necessary that we will be deep friends. We have correlation, but we do not know up to what strength our friendship is. So similarly, to judge the strength of correlation, we need to work out certain measure. And that measure is correlation coefficient. So correlation coefficient will tell us the strength of correlation. So now, next is your regression analysis. So friends, just now I told you that correlation just tells us qualitative answer. Okay, we are very close friend, where we are very deep friend, but it cannot quantify. It cannot quantify that up to what percentage out of 100 we are having friendship. Is it 99% friendship or say 50% friendship or 20% friendship? We don't know. So if we want to quantify this relationship, correlation relationship, then we need to go to regression analysis. If we want our answers in mathematical forms, then the correlation data need to go for regression analysis. So now see what regression analysis is doing. It collecting the data on dependent and independent variable. First we collect the data, then plot the data to visualize relation between variables. Then we will plot and make the scatter plot. What kind of relationship these variables are having? Is it linear? Is it quadratic? Is it cubic? Is it exponential? So it will give us some idea that what is the nature of the relationship, and then determine the strength and direction of relationship using correlation coefficient. So first we'll see whether it is positive correlation or negative correlation and how much closely these two variables are related. That can be just by correlation coefficient. Okay. Now we will go for fitting a regression line by least square regression or least square method. I have shown you that line is also called as regression line. The line which we are fitting is also called as response curve. You can also call it regression line. It has same meaning. Now, once we have fitted the line, now we want to see whether this line is significant or not, whether it is genuinely linear or not, whether it is genuinely curvilinear or not. For this also, we have got certain tests. So now assessing goodness of fit of regression line using R squared and standard error of estimate. So here we can find out the value of coefficient of determination. Friends, there are two R. One R is small r, simple R, that is called as correlation coefficient. But you will have R square. R square is called as coefficient of determination. I would explain to you in the later part what is difference between correlation coefficient and R square values. Now using regression line to make predictions. Now whatever regression line we have made, maybe a straight line or curvilinear. we need to test it for its significance. If it is significant, then we can make predictions. We can predict the values of dependent variable based upon the values of independent variable. Now, regression analysis. What actually we do in the regression analysis? In the last, we have seen what are the steps in regression analysis. So what is regression analysis? It describes the effect of one or more variable that could be independent variable on a single variable that is dependent variable. For example, here can be two independent variables. One can be nitrogen, other can be phosphorus. One can be nitrogen, other can be herbicide doses. So there can be two independent variables, and one dependent variable, we can take grain yield, we can take plant height, we can take any one on a single variable by expressing the latter as a function of the y former. So y is a function of x. This is regression. In experiments on yield response to nitrogen, yield is dependent variable and nitrogen rate is independent variable. Regression analysis is a method for investigating functional relationship among variables. Here, functional means quantitative. Quantitative means it is mathematical kind of relationship. This relationship is in quantities. That 1 kg nitrogen is giving you 10 kg of grain. So this is quantitative relationship. If I say 1 kg nitrogen increases yield, so this is qualitative relationship. If I say 1 kg nitrogen gives you 10 kg yield, so this is quantitative relationship. So in regression analysis is a kind of, it give us results on quantitative relationship. The process of finding a mathematical model, an equation that fits the data is regression analysis. So through regression analysis, through regression procedures, we can find out the best fit line, which is method of least square. Now, friends, just some terminology before we go deep into the subject. Dependent and independent variable. The variable to be predicted, that variable which we are going to model or predict is called as dependent variable. Dependent variable is also called as response variable. Similarly, variable used to predict or model y are called as independent variable and are denoted by symbol x1, x2, x3. These variables are also called as predictor or explanatory variable. So you can see synonyms for dependent. Synonym word is response variable. For independent variable, it can be predictor or explanatory variable. The dependent variable can also be called as response variable, and independent as or effect variable, and independent as cause variable, reason. A correlation exists between two variables when one of them is related to other in some way. This is definition of correlation. Then a scatter plot or a scatter diagram is a graph in which paired sample data are plotted. Plotted means the data is put on the graph with a horizontal x-axis and a vertical y-axis. Each individual pair is plotted as a single point. You know it better than me because your algebra should be good. Now see scatter plot, how it is made. So different pairs are plotted. So there are two different variables. One is shown by blue color and other is shown by orange color. So just you see, suppose blue color, series 1. So the data start here, 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 here. This is blue data. And then you can see orange series. So one could be nitrogen, other could be phosphorus, related to phosphorus. On x-axis, you have independent variable, and here you have dependent variable. So you can see here that this blue is showing up to some level, it is showing linear relationship. But then there is fall. And then again, roughly linear relationship. But there is very big fall here. If you see the orange data, orange data move like this. It will go here, then here, then here, then here, then here, then here, then. So you can see some trend is there in the data that we need to judge through our visually, that we need to judge through our naked eyes. The linear correlation coefficient measures the strength of linear association between the paired X and Y quantitative values in a sample. So to know the strength of correlation, we measure or calculate linear correlation coefficient. It is also called as Pearson. The full name is Pearson product movement correlation coefficient, in honor of Carl Pearson. Carl Pearson was mathematician who developed this correlation coefficient, or who gave the formula first time to calculate the correlation coefficient. He developed it. Simple and multiple correlation and regression analysis. Friends, what is meant by simple correlation analysis, simple regression analysis, multiple correlation analysis, and multiple regression analysis? There are four words or four terminology. If one independent variable affect other dependent variable, one independent variable affect one dependent variable, and you are doing correlation analysis, so this will be called as simple correlation analysis. If one independent variable is affecting only one dependent variable and you are doing regression analysis, this will be called as simple regression analysis. If two or more independent variable are affecting one dependent variable, remember, two or more independent variable are affecting one dependent variable, then this can be called as simple correlation analysis, or multiple correlation analysis and multiple regression analysis. So this is how we define the multiple correlation analysis and simple correlation analysis. In simple relations, only two variables are there. In multiple relations, you have two or more independent variable and one dependent variable. So now see the difference in correlation and regression. Now see positive correlation. Consider the following saying: the bigger they are, the harder they fall. The more the merrier. Easy come, easy go. The better the deed. Means in all these sayings, you are saying that one is affecting the other. You can study it later. So there are some other proverbs. These are called as proverbs. Take another group of sayings. Much haste, little speed. Small pitches have big ears. The best gift comes in small packages. So they are showing negative kind of correlation. In another case, you have seen positive kind of correlation. So you see positive correlation means one variable is increasing, other is also increasing. So this kind of relationship shows us positive correlation. So these data are scattered. Similarly, negative correlation, one variable is increasing and other is decreasing. This is called as negative correlation. There may be a situation when both variables are decreasing. There may be a situation when both variables are decreasing. Such relationship is called as positive correlation. Now you can see more positive correlation. So there may be some non-linear relation or no linear relation. So you can see this is scatter plot. No any trend is shown. A relationship between two variables for which point in corresponding scatter plot do not fall in approximately a straight line. So you cannot make a straight line from these points. Therefore, this shows you no correlation. And then regression. What is regression? So regression is amount of change in one variable associated with a unit change in other variable. If you make one unit change in X, how many unit changes are there in the Y? So we are understanding the quantitative relationship. If we change one quantity of independent variable, how much quantity of y variable is changing. That is regression. Correlation refers to the fact that two variables are related and closeness. However, regression, on the other hand, refers to the nature of relationship. Nature of relationship, and see the following sayings. You can see the following sayings. A penny saved is a penny earned. Means you have saved one Afghani rupee, and that means you have earned one Afghani rupee. If you have saved one Afghani rupee, you have saved one Afghani rupee. You have earned one Afghani rupee or penny. A bird in the hand is worth two in the bush. So you see, numerical relationship is there. One bird is in your hand and two birds are in the bush. A stitch in time saves 9. So you see mathematical relation, quantitative relation. One picture is worth 1,000 words. Now we can make a table out of this. So one is independent variable, other is dependent variable, and regression equation will be like this. And what will be the regression coefficient? The last column is regression coefficient. First column, pennies saved and pennies earned. You remember, one penny was saved. If you have saved one penny, that means you have earned one penny. So it means 1y is equal to 1x. So regression coefficient will be y is equal to 1x. So 1 is multiplied to this x. So 1. Then one bird in hand, worth 2 in bush. So there is mathematical relationship. So y is equal to 2x. One bird in hand and two bird in bush. So y is equal to 2x. So this 2 is regression coefficient. Stitches in time. So here one stitch is time saves 9. So here is the saving. So y is equal to 9x. So 9 is the regression coefficient. And one picture is 1,000 words. So y is equal to 1,000x. So this is actually regression, mathematical relation. And you see it is 1,000 times. 1,000 times better. A picture is 1,000 times better than words. Picture is better than 1,000 words. So you get 1,000. So these 1 to 9,000 are regression coefficient. Regression coefficient and intercept. Friends, let us go to the last link, link number 4. I wish to take only 10 minutes more, 10-15 minutes, and you will not have any class this afternoon. It is an announcement because you got namaz and some religious work. So therefore, today you will not have any class in the afternoon. Can I take class for 15 minutes more or not?
Okay, sir. Okay, sir. Please continue
Okay.
it.
So
We will
join
go
link.
to the 4th link.
Okay, good. Fine.
Okay, sir. We will join on 4th link.
Yeah, good. So welcome again, and you have seen the difference in regression coefficient and correlation coefficient. So here regression coefficient is how many times this y, how many times we should multiply to y, to x to get y. That is your regression coefficient. Now you see this general equation for a straight line is y is equal to a plus bx. Now try to understand the meaning of a and b. So in this case, if we can find out the value of a and b, that means we will find out the linear equation. So simple. You need not to measure the distances, do that much of things. But if you can find out value of a and b, then you can put those values here and you can develop the equation. So the symbol a is called as the intercept. Here y is equal to a plus bx, a is intercept. Intercept means the value of y when x is 0. The value of y when x is 0 or x is control, that value of y is called as intercept. It is when x is 0. So y is equal to a. Hence the line crosses the y-axis at units from the origin. This line will start from the a. This line will start from the a. When a is 0, the line will start from the point of origin. You could understand. Now regression coefficient and intercept continues. The symbol b is called as the slope since it determines the steepness of the line. So p is regression coefficient, means if you make a unit change in x, how many times unit changes are there in y? So this is regression coefficient. which is called as B here. So it is easy to see that B is the amount of change in Y associated with a unit change in X. Therefore, it is logical to call B as the regression coefficient. B is called as regression coefficient. If you have quadratic equation, in quadratic equation, you will have C also. So B and C will be the regression coefficient. In cubic equation, this B, C, and D will be the regression coefficient. There you will have 3 regression coefficient. In quadratic equation, you will have 2 regression coefficients. Linear relationship, try to understand the relations between 2 variables is linear. I think I will continue from here, from linear relationship in the next lecture. I will start from here, this point, so that linear relation is covered in full. And I don't want to take your much time because you are getting late to namaz or your other work. So you can show me your faces if you have video. If you can put videos on, we can say just discuss for 1 minute. So you are all teachers there or students? You are muted. You are muted. Open your voice.
Oh, sorry, apologize, sir. So our class,
we are
so class students has different jobs. I am working in a national NGO as a South regional directors main positions, and have three projects and working in an NGO since 12 or 13 years. I have working experience in the NGOs, in international and national NGOs. So we will give chance to another friends.
Good. Thank you. Congratulations. You are doing good job. And Hikmatullah or Ijatullah. Ijatullah. Hikmatullah.
In the name of Allah, I am Izzatullah.
This is Said Hikmatullah, sir, and I'm also one of the classmates. I'm not a teacher. I'm working in local NGOs also.
Okay.
So
Yeah. I guided long back. I guided one MSc student in IRA. Muhammad Amir. My student, regular student, was there. He came here for 2 years and published good paper. I'll send his detail also. Amir or Amir. And his surname was Dalwa Zai, Delveta Zai, something like this. In the end, it was Zai, Z-A-I, Zai. So he came here and he came 2-3 times to India. One time he brought his uncle for treatment, medical treatment. He had some problem in his spinal. So he visited me every time whenever. Amir Jan. So, and you, Rahimullah, you are a teacher or a student?
Thank you, sir. No, I am now a student, and also last few years I worked with NGOs here in Afghanistan, and also in 2013 I visited from India, especially in Bangalore area. Also, I went to.
Actually, you can try. One fellowship is there. One fellowship is there in India. You can come here as a regular student. If you want, you send me email. I will send you detail. Some of you can apply for fellowship. And some Afghanistan
Inshallah.
are going to China also. You can apply for Chinese fellowship also. And many countries would welcome you. China, India, even Russia.
So when the master degree finished here in Afghanistan, India, after in Doctora will be go there beside of you will learn much things, many things, sir.
So Nabi, you want to say something, Nabi and your friend or anybody else?
Yeah, thank you, teacher. I'm Muhammad Nabi. I'm an assistant professor in one of university in Afghanistan.
And your friend sitting beside you,
In the name of Allah, my name is Muhammad Akbar. I am an assistant professor in Afghanistan, one of the university.
good. And one person is left, he did not introduce.
Actually, 2% is remaining, sir. One is Izzatullah. He is escaping, I think, in that trackings. And also we have Kadir. Izzatullah
then.
is sitting.
The class
Izzatullah is
is,
sitting beside Hikmatullah.
Wish you all
So
the
Izzatullah,
best. Wish you all the best. And tomorrow is Saturday. Saturday, Dr. Y.S. Shime will take your class and he will send you the link. And it will be only in forenoon. Tomorrow is Saturday, forenoon. He wishes to take your class. He will send you the links. Okay.
thanks.
This was
Thank you, sir.
information. And today afternoon, there is no class. Okay.
okay, sir. Okay, sir.
Bye.
Thank you
Bye.
very much. Bye, bye, bye.
Bye bye, sir.
وليجي خبرتك ولي ذاتها.